All right. And since we do have such a large crew, I am going to forego introductions. It is great to see so many faces here and so many people that turned out to the meeting. Hopefully many of you know each other. And if not, hopefully we'll have some time down the road to get a little bit more face time. But thanks again for joining us today. And I'm going to be primarily guiding the presentation, and Scott is going to be keeping an eye on the waiting room and also the chat. So certainly if anything comes up or if you need to get in touch, something's having any issues from kind of the tech side, just shoot Scott a chat message. So with that, we're going to go ahead and get started. And I did want to make this a little more interactive than a lot of our Zooms are lately. So I don't know how many of you used Slido before, but it is actually kind of a nice tool when you're working with a large group and you want to be able to kind of solicit some information. And so if you can, either you can use your phone and take a picture of that QR code and it's going to pop you right to a screen. There's no download or anything like that, but you'll be able to interact with the meeting a little bit better. Or you can actually just open another web browser and put in slido.com, and then it's just going to prompt you for a number, and that's the Slido number there. So I'll give you guys a little bit of time to do that, and I'm actually going to advance the slide. It'll have that information on there still on how to link up, but wanted to start with a question. So first of all, just what is your affiliation? Really, this is just to test the tool more than anything else. So go ahead and you can drop in what your affiliation kind of with this meeting is. And certainly if you represent a couple of these, go ahead and answer accordingly. Kind of nice to see who all is in the room with us today. And I also wanted to point out one other feature of Slido. There is actually a Q&A piece to this platform, both on your phone or if you pulled up a web browser on your computer, you should be able to drop in questions in there, and we can actually rank questions. So when we get to kind of the more of the discussion part, that's a really good place to drop questions and see if other people maybe have the same one. So feel free to use that, or we'll also be monitoring the chat too. So give it just a little bit more time. Still got some people actually coming in the room.
Ashley, if you make me co-host, I can help let people in.
Oh, sorry. Yep. Was planned to do that early on, and then when I didn't see you, I forgot to pick that up. So do that. There you go, Scott.
Good afternoon.
Hello, hello. So if you just were able to join us, feel free to connect through our Slido app, either on your phone or in a web browser. You can use the QR code or the URL there. And we're just doing a quick polling just to see who is on today for our Barley Pest Initiative virtual forum. All right, so we got a good mix here, certainly dominated by our BPI researchers that are receiving funding, but we also have some folks that are researchers that aren't. And then very cool to see some folks from the National Barley Improvement Committee, the individuals that have been busy lobbying to get additional funding for this work. And then we've got some administrators and even some members of AMBA on. So thanks to everyone that humored me and is using the poll. We'll have a couple other questions throughout the presentation too, but thanks a lot for doing this, because it is a way to make it a little bit more engaging and also get a better clue of who's all with us today. So, all right, you can keep actually answering that, but I'm going to go ahead and click off now and advance through my slides. So the Barley Pest Initiative, one thing I just wanted to start with is just kind of providing a little bit of context for people that might not be as familiar with the initiative. It was established and was first received funding in FY21, and we'll talk a little bit about that funding history. But the main purpose really for the BPI was to target about 20 different pests outside of Fusarium head blight, knowing that the U.S. Wheat and Barley Scab Initiative was very focused on addressing that issue, and that the Barley Pest Initiative that was developed through work by Mike Davis, my predecessor, was really to try to address all of these other insects and disease challenges that we have in the barley crop. And so to do that, really, there was two parts, primarily, and where most of the funding lies right now, to try to identify new resistant varieties. And so a lot of the funding does go to breeding programs, but also support research around those breeding programs. And then also there was a mention in the BPI of doing some development of management strategies to really mitigate those impacts of those pests as well. So this is really what the kind of the Barley Pest Initiative is based on. I know many of you already know this and understand these talking points, but for those of you that might be relatively new to the initiative, I just wanted to share that. So by the numbers, and so I'll talk a little bit more about the questionnaire that we did last summer, but I wanted to just, again, to provide a little bit of context to show how broad-reaching this initiative is. And so the list down the middle shows all the ARS facilities and then our university partners that are actively working on the BPI. And through the questionnaire, we were able to identify work happening in 15 different states that actually included over 50 researchers. And so for each of these facilities, there's a unique person identified as kind of the lead in this initiative, but then there's a lot of other research partners throughout the country and even into Canada that had some involvement in some of this work. And so a big thank you to all of you for reaching out and really broadening this research network so that we can get the most work done. So really exciting to see that many people engaged in this work. And then the other exciting piece to this was that the BPI funded research led to over 20 different publications. And I know that some of the publications would have likely happened without the BPI funding. However, it's really exciting to know that the BPI funding did directly go towards the advancement of science in this area and on these pests. And so I'm really excited to kind of get that kind of data point as well. And a big thank you to everyone that reported those in the questionnaire. Certainly, if there's publications that are coming out in the future, or maybe there's things that have happened that you didn't get caught in that last questionnaire, that is something I do hope to do on an annual basis. And so we'll be able to catch those later. But thanks to everyone that reported those, because I thought that was another really nice data point that we can share. And this information is extremely important as we go back to Congress and ask for additional funds. The more states that we can talk about in our story and who we're reaching is going to allow us to actually get into more offices and to tell the story about what the BPI is doing. So it's really exciting to see that we've got such great reach. But also when we're in there telling the story and talking about this part here about actually getting publicized research out of this funding is another really nice story that we can tell Congress and ask for additional funds. So just a quick snapshot on the actual funding progress of the Barley Pest Initiative. And so this chart here just shows how we received our first 1 million in FY21, our second million in FY22, and then right before the holidays found out that we did receive a third million in FY23. And keep in mind, this is annual funding. So each year, essentially that appropriated funding level has gone up incrementally. And this nice steep line of the curve is hypothetical. So we would like to get to 5.3 million eventually. And that is what we will be actively lobbying for when we go back to Congress next month. And so we've got a bit of a reach here. We've been getting about 1 million each year leading up until now. And so we're at that kind of point now where we're just trying to capture that last 2.3 million to kind of wrap things up. So we'll see if we get there, but hoping for at least an additional incremental adjustment to this so that we can keep funding that research. And like I mentioned, the National Barley Improvement Committee will be returning to DC next month to ask for that increased funding. And I should note too that that's actually our primary ask of our lobbying effort this year, is that we will be actively seeking funding and trying to get to that 5.3 million. So I did want to just say thank you to everyone that took time to fill out the BPI questionnaire. I know that that was something relatively new for folks, at least through this initiative. And there was really no kind of mechanism to understand the work that was happening through the BPI. And so I felt it was really important for us to be able to kind of do a snapshot, benchmark the people that were working and what they were doing, and also really to kind of have some accountability of the pests that we said we were going to work on and whether or not we actually are working on those. It also allows us to tell much better stories in Congress, and that's why I asked you all to provide very kind of short, brief reports back to me, because that's what I can use then when we're in these offices talking about why the BPI is so important. After this meeting, I will actually be sending a follow-up email with this recording, and also just a very quick survey on this virtual forum. But I also will be sending along a PDF document of this annual report. So you can see a picture here on the screen. But what I did is I condensed down most of that BPI questionnaire data that I collected and actually put it in a report form. It provides very brief snippets of the research that's happening at all the various locations, but also provides some of the more kind of generalized summary data that I'm going to be sharing with you today on what pests are currently being addressed and what work is happening and what work is planned for the future. So hopefully you can use this document to also help inform your stakeholders of the work you're doing. And I would love to hear any feedback you might have on it, on how we can make it better and that you can use it more for a tool for yourself so that we can continue to build support around the Barley Pest Initiative. So now I would like to move into some what I'm calling lightning presentations. And I want to say thank you to everyone that agreed to do these for me. No hurt feelings if you didn't get asked. I literally just kind of randomly grabbed a couple of our ARS sites and a few of our land-grant universities and asked them to just very briefly share what they're doing. Again, this is more for some of the folks that are joining us today that maybe might not be as familiar with the BPI, but would also just like to hear some of the research that's happening. And so with that, I'm actually going to go ahead and get started just so that we try to maximize our time. And we're going to be starting with ARS Aberdeen first. And just keep in mind too that even though we have one speaker for each of these locations talking today, that they certainly represent a much larger team that I'm sure they're going to share as well. So with that, I'll turn it over.
Hello, everyone. My name is Dong Yinggo, and I'm a plant genetics and the USDA ARS small green and potato germ plant research unit. There are 4 research groups in this unit. For the barley and the oat group, we have 3 scientists. Gongxia's team focus on the developing new barley cultivars for multi and feed use. Casey's works on oat crop rust and the molecular mapping of some important traits in barley and oat. My lab works on the barley genetics, germplasm enhancement, and improvement of barley FHB through the RNR technology. We also have National Small Grains Collection Group. They maintain and distribute the seeds of wheat, barley, oat, rice, and other small grains to the research in the United States and other countries. Please, next slide. Next slide, please.
Oh, is it not advancing?
Yeah, okay, okay.
Do you see it now,
Yeah.
the one that says the work we have done at the top?
Yes, these
Okay.
is these slides. Okay, these slides I just show you some highlights of my research data. Or unit data, because I just joined the unit in February 2021, and I didn't get much time to work on the barley pest and related project. But my colleagues and the private scientists and our collaborators, they did a fantastic job on some on some barley strip rust. But first, we developed some barley germ plants that showed a good resistance to BSR and seedling and field stages. We also develop several genetic population and mapping some cultures related to with BSR resistance in GZ and 95 SR360A. But then we also conduct some parameter breeding work because the GZ, this is the germ plant which show wide spectrum to BSR, but this germ plant which has very poor agronomic traits. We make a cross between GZ and 95 SR360A and develop some lines which show the good BSR resistance and better agronomic traits. We also found some linkage between the major culture on chromosome 4 in GZ with powdery mildew resistance and low yield drag. Okay, please go next slides. This slide is some preliminary plan or future work. But first, we plan to identify new BSR resistant resource in wild barley, especially. We collaborate with the National Small Grain Collection Group, and we already genotype over 1,900 accessions of wild barley with the 50K SNP array. With this genotype data, we will generate a molecular RD for each wild accessions. Also, we can identify some duplicate accessions. Next step, we will also screen the BSR resistance in these wild barley accessions and mapping the BSR resistance in the wild barley accessions. We will also continue our work on developing new BSR resistant barley cultivars or germ plasma. Also, we would like to also to collaborate with any group to other disease and pest, because in Idaho, actually in here, we have very low disease and pest pressure, but we didn't see too much disease and pest in our field, but we just found the aphids in last year. It's very looks serious. Yeah, thank you so much.
Sorry about that. Couldn't get to the mute fast enough. Thank you very much. And just for everyone's knowledge, we'll go ahead and do Q&A at the end of these presentations, just to make sure we stay on schedule. So next up is Montana State. And Jamie, are you still on with us?
Yeah, I'm here.
Awesome, thank you.
Okay, so one of the main things that we've been doing is just making crosses for known resistance genes. As you all know, in Montana, disease doesn't tend to be a problem as much because we're so dry, but we want to be ready in case there is a problem. And so by working on these crosses now, hopefully we'll have something in the future. Next slide. Importantly, we're doing that with the winter program as well. And thanks to our collaborators, these lines are being evaluated where they need to be evaluated. And a result of that is that we have identified some stripe rust resistant buzz lines. Right now, we're moving forward with those, testing their malt quality, hoping that they will have good malt quality and eventually they will be released. Next slide. Of course, that trial data is being used to help inform further crosses to make more resistant lines. And then we focused on a bacterial leaf streak some because we noticed a complex set of leaf diseases in previous years, especially in irrigation. And from that work, we at least one of the components of that disease complex is bacterial leaf streak. And a microbiologist here at MSU, Qing Yan, isolated 2 different strains from our barley samples. He was working with one of my students, and using ribosomal RNA, he identified those as Xanthomonas translucens. And we've confirmed the pathogenicity in a greenhouse inoculation. And so mainly just to get an idea of what's causing the problems here in this disease complex. And then we've started looking for resistance in our own material. And we've been collaborating with Molson Coors on this as well. They've been evaluating some of our material, and we've been evaluating some of them. One of our big problems is that we don't get consistent disease pressure. Next slide. One of the concerns, particularly as we build the winter program, is it's been previously reported that leaf streak can increase if you have ice. And so here's just a picture of a recent winter field. The brown you see there isn't disease, it's death due to cold. So we're, that's our primary focus, is try to get increased cold tolerance, but we're also looking for Xanthomonas pressure in this material that's being exposed to freeze thaw. Next slide. A unique thing about Montana is that we've had consistent wheat stem sulfide pressure. Originally, people reported barley to be very resistant, but recently we've found that there's variation in wheat stem sulfide pressure across varieties. And so we view this as both an opportunity and a threat. The threat is that we are seeing an increased level of cutting of barley lines this year. There was a report that a lot of our malt lines that are currently being grown were cut heavily this year. And when a line is cut by the sulfide, then it essentially makes it impossible for the grower to harvest it. It also decreases yield and quality. And so it's a problem that's increasing. The cool thing is that we do have some resistance. Unfortunately, it's in a line that isn't a great malt line, the line that was released named Craft. And so we're looking towards identifying what that resistance is and then using it to improve our resistance. The other thing that's cool about this is that it plays over into wheat. For a long time before I became the barley breeder, I worked on wheat stem sulfide resistance in wheat, and that's primarily due to solid stems. This resistance has nothing to do with solid stems. These stems are not solid. So it could be a new form of resistance that potentially could be taken advantage of by more than just barley. And I think that does it for me. Thanks, everybody.
Amy. I know it's really hard for our researchers to talk about everything they've done over the last couple of years in 5 minutes, but I really appreciate everyone doing that. And next up is Oregon State. I think we've got Bridget here to present for that team.
Great, thanks, Ashley. So I am representing Kat Hayes' lab at Oregon State University. And this work that I'm going to be talking about also is being done with a postdoc, Chris Massman, who I believe is on the call, and then Dr. Christina Haggerty, who is our cereal pathologist over on the east side of the state. So I'm just going to talk specifically about a few of the projects that we're working on. And so one of these is looking at resistance to barley yellow dwarf virus, which is becoming more problematic with changing climates and temperatures that aren't dropping as early as they used to. And so BYDV can cause severe crop losses. There are a number of viruses that are transmitted by many different aphid species that can result in this disease. And currently it's quite difficult to do an inoculated experiment because raising aphids is very difficult and inoculating lines is very difficult. And so we decided to take advantage of natural pressure in the field and planted probably 3 or 4 weeks earlier than we usually do to make sure that we got enough pressure, and planted out this large, what we call cycle 6 elite malting panel, both in Corvallis and then working with collaborators at UC Davis. And so the goals of this project are both to identify genetic determinants of resistance using this large GWAS population. And then we're working with some USDA entomologists and pathologists here in Corvallis to characterize vector and virus populations and dynamics. And then also to just assess the feasibility of, using non-inoculated germplasm to do the phenotyping to see if that's even feasible. And we're also working with another researcher here at OSU to do remote sensing and assess these fields with drones. And so those are the images that you can see here. So this is the first official year of the experiment. Last year, we had quite a bit of pressure and took advantage of that to create this cycle 6 population. And so this year is the first year that we're really working with some of these USDA collaborators. And so stay tuned for the results of the GWAS. Yeah, great. Next slide. And then another project that we're working on is investigating scald and looking at the barley leaf microbiome. And so scald is a disease that's predominantly important for winter barley, which is the main focus of our breeding program here. And although there have been a number of identified QTLs for resistance, most of those have not been specifically used in breeding. The disease is also very environmentally dependent. And then we've also found that maturity and heading date really confounds the resistance. And so this is a biparental population that was created by a cross of Lightning, which is one of our malting barleys, by Survivor, which is a spring line out of WSU that is IMI tolerant, so it can be used after production of Clearfield wheat. And so this population has been grown in the field and then genotyped with the 50K and then phenotyped for both scald and heading date to sort of try and parse out scald resistance from the heading date QTL that we've identified. And then we're also looking at the leaf microbiome composition in collaboration with the Posey Busby lab in our Department of Botany and Plant Pathology, and that work is ongoing, but we're very excited to see the results from that. Next slide. And then our work with Dr. Christina Haggerty over on the east side of the state. She works predominantly on dryland wheat, but has been using barley as a cover crop in different trials to help reduce the presence of soil-borne fungi that are problematic for both wheat and barley, and has been seeing some exciting results from that, looking at barley as well as a few other different cover crops, and has seen that using barley as a cover crop can help break these cycles for wheat. So as Jamie mentioned, you know, this research has broader implications than just the barley community. And I think with that, that's all I've got.
Thank you so much, Bridget, and to the rest of the team at Oregon State. Next up, we've got the University of Minnesota. Brian, are you on?
Yes, I am.
And I'm sorry, your slides are a surprise.
Yeah, for those of you who want the unabridged version, I can send you a longer version, but I'll work from just a couple of slides here. But just very briefly, unlike the drier areas like Montana, we have quite a few diseases that we have to worry about in Minnesota. If I were to rank them, those top three probably are the ones that we fear most year in and year out. But the two listed in green, spot blotch and stem rust, you're reminded how devastating they can be if you have susceptible materials. So we sometimes forget about those, but we have to make sure that we have resistance to those as well. And then with Kevin Smith's program, he's got a very diverse barley breeding program that consists of spring type, facultative types for with winter hardiness as they're sown in the fall, and also has a program with naked barley and organic barley and so forth. So we have quite a few germplasm to deal with regards to screening for disease resistance. But what I would just like to say, just very briefly, is that with Kevin Smith's material, we're screening anywhere from about 400 to 800 lines per year. And we like to do these assessments in the field if we can. So we're doing primarily net blotch, bacterial leaf streak, and spot blotch. And we also do stem rust as well. And that is considering both domestic races, but also some of the African races as well. We still worry quite a bit about stem rust because although we've had durable resistance provided by RPG1, we know that there are some domestic races like QCC that came up. And then if you speak with Robert Brueggemann's group at Washington State University, he has a number of isolates that are potentially coming off the sexual host Mahonia or barberry that can overcome a lot of the resistances. So we still need to really look in detail about providing evaluation and screening for those. Some of the other sort of incidental diseases that we see are powdery mildew, leaf rust, BYDV, septoria, and scald primarily. So we may not screen all of these every year, but we try to do them every so often before Kevin approaches more of a later term a selection for plant scale or pilot scale. With regards to the group, I should mention as far as the group at Minnesota, this project is led by Matt Rouse and myself, but we also have collaborators, of course, with Kevin Smith, the barley breeder, Gary Milbauer, Ruth Delmacki, and we also have two postdocs, Ahmed Salam and Eric Nazarino. So from the yearly screening data that we have, we try to work on characterizing the genetic architecture of disease resistance. So the great part about Kevin Smith's program is he basically has every one of the breeding lines that he has in his program genotyped with 50K. So we're running these data past our phenotyping data to provide information on where the resistance loci are located and also their allelic effects so that we can feed that to Kevin's program in a genomic selection program. So this is being done basically for about 5 or 6 different diseases. We wish to add more of those, particularly when we think about some of the winter barleys may start to succumb to diseases like powdery mildew and leaf rust. I wanted to mention just one other thing about leaf rust. Through Matt Rouse's postdoc, Eric Nazarino, he has a national presence in terms of conducting national surveys for Puccinia hortii virulence, and also a national footprint in terms of screening some of the breeding program's materials against the leaf rust pathogen. So Eric is doing work on trying to do gene postulations to provide that information for breeders to keep the good genes into their program. So that's a very quick nutshell approach. I guess as far as challenges, I would say for us is that the different germplasms that Kevin has, so we can't guarantee that we have the same package of resistance genes in all these different germplasms. So that's a little bit of a challenge. We also are concerned about new virulence types that pop up in pathogen populations. For example, we've had durable resistance, as I said, in stem rust and also spot blotch, but there was, in addition to the QCC stem rust, we also had an isolate pop up in North Dakota that was virulent on our durable resistance. So our program will continue to identify new sources of resistance and characterize the genetic basis. And as many people who are familiar with my program, I do a lot of work with wild barley, so we're definitely tapping into that. And I think also, although in Minnesota we have a good opportunity to screen for a lot of these different diseases, there are some diseases that just typically don't occur with high frequency, and we can't work with them so easily in the state. So it would be great to collaborate with other folks as well, and we would be happy to reciprocate as well. So that's it in a nutshell. Ashley, thanks for the opportunity.
Yeah, thank you, Brian. Thank you very much. We were kind of chuckling before because he sent me a few too many slides, so it was going to be a total surprise which ones I actually put on the screen. All right, so one more presentation. Hopefully you guys are getting a good flavor for some of the work that's happening out in the field with the BPI. So now we're moving towards Fargo.
All right, thanks, Ashley.
Tim. Yeah.
So, so my lab is half wheat, half barley. So, and I'm the lead on the pathology, Chris, here in Fargo. So I'll just talk a little bit about one of the diseases that we work on. I work on both net form net blotch and spot form net blotch, focusing mostly on the pathogen side, pathogen genetics and effective biology. So this is a project that was just recently completed by a grad student in my lab, focusing on the P. terry's maculata and the barley susceptibility resistance, and focusing on genetics. So we use both host and pathogen genetics. And if you can see up here in the upper right-hand corner, we crossed the line Hocket, which is a Montana susceptible for spot form net blotch, with a PI line PI 67381, which is resistant to both spot form net blotch and net form net blotch. And you can see we have two major associations, two regions, one on chromosome 2H, one on 7H, that pop up in the QTL analysis. So we crossed this PA14 isolate, which is virulent on Hocket, with a CAWB isolate that stands for California wild barley. These are isolates that are not virulent on domesticated barley. So when we cross them, you essentially have a virulence allele at all of the virulence loci that come from PA14. And you can see that we have two associations, that pop up here. One on chromosome 1, which it looks like it's been trimmed off on the bottom of the screen, chromosome 1 and chromosome 2. So the next question that we had was, does chromosome 1 and chromosome 2 target the chromosome 2H and 7H? So the first thing that we did was look at F2 populations of the Hocket by 67381 to show that both the 2H and 7H appear to be dominant susceptibilities rather than dominant resistances, meaning that the pathogen is probably producing a virulence factor, possibly a necrotrophic effector that's targeting these loci. So if you go to the next screen, So we divided the population into 4 genotypes. On the left side here, you can see in red the genotypes on chromosome 1. So the top group here has the virulence genotype on chromosome 1 and chromosome 2. The second group has the avirulent or lacks the virulence on chromosome 1, but has the virulence on chromosome 2. The next group, the opposite, it has the virulence on chromosome 1, but lacks it on chromosome 2. And then the last genotypic group lacked both virulences. So when we inoculate these groups or representatives of these groups across this Hocket by PI 67381 population, you can see if both PA14 virulence alleles are present, we get both QTL, the one on the 2H as well as the one on the 7H. So that's in black on the top panel. If you only have the chromosome 2 virulence, you only have an association at 7H, meaning that this chromosome 2 is targeting the 7H locus. And if you look at the genotypic group that has only the PA14 on chromosome 1, you can see that you only get an association on chromosome 2H. If you eliminate both of them, you have no association on either of those. So we're showing here, the indication is here that PA14 has virulence factors that are targeting susceptibility, like other necrotrophic pathogens on this 2H and 7H locus. Currently, we're looking at the effectors. So to clone and characterize the effectors that are underlying these QTL, Shengming Yang's group is working at identifying the susceptibility genes that underlie these loci in barley on chromosome 2H and 7H. So that's the direction that we're headed, and we still have a grad student that's continuing to work on, actually 2 graduate students, one in Shengming's group and one in my group. So go ahead and go to the, yeah, there we go. So additionally, Jeff Schachterly is working in my CRIS on bacterial leaf streak. And he's developing genetic and disease screening tools to better understand the BLS on barley. So he's in the process of developing tools so that we can do continued evaluation, especially at the molecular level of what's happening in this bacterial leaf streak. So if you want to know more about that, you can contact Jeff directly.
Awesome. Thanks, Tim. And thanks to everyone that made presentations today. Again, I know that this isn't an exhaustive report on everything that's happened over the last couple of years, but hopefully it's a bit of a teaser just to kind of show some of the work that's happening. And I'd love to talk later during this forum too about whether or not a more in-depth research kind of report out is necessary, or if maybe this information is getting already distributed widely in other platforms. So we'll talk about that a little bit later, but want to just thank those that did prepare these presentations and for keeping them so brief. So we kept pretty good to time. So now I want to kind of dive in and actually start talking about the work that has happened over the last 2 years, the first 2 years of funding. But first, I just want to do one more of these Slido polls again, just to try to keep people engaged in the conversation. I know that some of us are kind of at the like post-lunch slump right now. So either you can get in through the QR code on your phone, or you can go on and open up a web browser to this Slido, but this is actually going to be a word cloud. And so essentially what we want you to do now is just to type in what you perceive to be the greatest pest, insect, or disease. And I love how it says other than FHB and fusarium's the first one pops up. And that's okay too. But hopefully as these things start popping up, the BLS and bacterial leaf streak are both up there. I see that. I knew that was going to happen. We'll probably get different versions, but they'll still all pop up. So it'll be nice to see what folks are thinking about, which ones they're most worried about right now. So keep going and typing those in. Because then what I want to do is I want to actually look at how the first 2 million really was distributed over the last couple of years based on our pests. So give it just a few more minutes to or a few more seconds to let people type. And thanks to everyone that's participating. like stripe rust is a big one. Bacterial leaf streak, or BLS is another one. Got a few bugs speckled in there every once in a while, but it's like the diseases are prevailing. All right, looks like entries are slowing down. One more person's typing. I'll let them get in, then I'll go ahead and close it. All the rust. That's a good response too. All right, gonna go ahead and close it. So it looks like stripe rust and BLS might be tied. What I'm seeing on this one, I will save this word cloud too and share it with you all, but thanks for participating and being a good sport on this. I'm going to go ahead and go to my next slide, which is actually going to show you how the funding was distributed over the last couple years. And so I'll tell you a little bit about how I got to this chart. So first of all, keep in mind that this questionnaire that we distributed was asking questions based on the last 2 years. And in that first year, there was 1 million distributed across all of the partners. And then on year 2, there was 2 million distributed across all the partners. So in total, we've had 3 million go towards this. And of course, we all know there's money that gets taken off the top. So it wasn't a total 3 million, but just in theory, that's how much was distributed. And so if you see on this chart here, stripe rust ended up receiving about 20% of the funds. And the way I came to this was in those questionnaires, identified which disease or insect was getting work done at each of the respective sites. And then I ran kind of an analysis based on how much money that location received. So this is weighted. It's not necessarily that 20% of the locations were working on stripe rust. This really, in theory, does look at how much money distributed to that particular pest. And so stripe rust was the one that received the most at 20%. And then after that, about 13.5% to leaf rust, 12% to BLS, and then powdery mildew there on the bottom at 11%, stem rust at 10%. And then the only bug that showed up in these kind of like top 8 categories was the green bug. And then net blotch and BYDV showed up on there too. So these are kind of the main pests that receive funding through the BPI. And considering one of the items in our word cloud was all the rust, that's good considering if you add up stripe, leaf, and stem, that definitely represents a pretty large chunk of the finances through the BPI and what was allocated. So also wanted to share with you all like what that other category looks like. And so these are about 11% of the funding allocations was distributed across these. And this was pretty small percentages. We're talking probably anywhere from like 2 to 5% each of these. These are very, very small. Some of them maybe even down to 1%. But these are the other pests that were identified. And then also wanted to highlight what I call the orphans. And so these are pests that were identified originally when the Barley Pest Initiative was originated. And these 5 items currently, at least through the questionnaire, no one mentioned working on. Not suggesting that anybody needs to be working on them either, but these are 5 that currently did not receive any attention, at least as reported through the questionnaire. So we've kind of got 3 categories here. We have like our top pests that people are working on. I'll go back to that one, the top, and I believe there's 8 on the screen here. And then, which is representing nearly 90% of the funding. This is representing about 11% of the funding. And then again, those orphans, the items that no one is currently addressing. So those are kind of like the three, you can say, categories of pests currently that we're looking at. And so what I want to do really next is actually open up some conversation about this. So I believe this is our last Slido poll in this presentation. What on this one you're going to do is actually, these are those top 8 that were shown in that first chart. I would love to just see everyone rank these and see whether or not they actually align with kind of how the funding was allocated. And then I want to open up into more of a discussion about that. So about 90% of the funding went to these kind of top 8 pests. And if you've got the Slido poll open either on your phone or in a web browser, you should be able to kind of shuffle these and rank them in terms of where do you think the need is highest. And I do want to just say, while folks are working on this, is that I fully recognize that some of these pests are getting funding from other sources. And so just because maybe there wasn't a higher amount funded through the BPI, I know that there's work happening beyond the BPI, and perhaps some of these diseases specifically are getting funding from elsewhere and really helping tackle those problems. And so maybe we don't need to be spending as much as the BPI, as much of the BPI on that pest. So just want to say that I do recognize that, and that's certainly at play here. But what I want to just make sure that we are reflecting on is whether or not we're currently being the most efficient with those resources through the BPI, and whether or not we need to maybe shuffle those priorities or think a little bit differently about how we're spending our time on them. So give it just a little bit more time for folks to respond to this one. I know not everyone probably feels prepared to answer this question, and that's totally fine, but I know that many of the researchers that are on the call today probably have some opinions about this. So this is how we maybe also start identifying some of these emerging pests that we're not currently working on that maybe we should be. So, all right. Things are slowing down with responses, so give it just a couple more seconds. All right. So there we are. So this is showing that stripe rust is currently, from the group that's on the call today, should be the top priority in terms of funding. And then we've got BLS next, and then leaf and stem rust, net blotch, powdery mildew, cereal cereal or barley yellow dwarf virus, and then that green bug aphid. So, all right. So that actually doesn't stray too far from what we currently show in the way that the funds are being allocated across all of the research. So that's actually a good thing that we're hitting the target pretty close there. So I guess what I would just like to open it up now is for discussion. So please, whether or not you want to drop a question in the Q&A through Slido, you can also drop ideas in the chat. But I also strongly encourage you to just go ahead and come off mute and share with the group whether or not you think right now our attention's focused on the right things. Do we need to be shifting kind of our funding priorities? How could we maybe be doing this work better? So with that, I just want to open it up and give some time for people to chat. But not all at once. I know you're all going to stay quiet there. So go ahead and start talking. Anybody willing to take that first shot at this?
Eric just wrote a note in the chat about whether powdery mildew could be a bigger problem in winter barley than spring barley. Yes, it possibly could. And then you also worry about the two crops being bridge hosts for one another in the infection. So yeah, definitely that can happen. I mean, we don't have it much on spring barley, but as we see more winter barley perhaps creep into our area, we might see that more of a problem.
All right, I'm going to also jump over to the Slido Q&A. Nicholas dropped in a question. Is variation to sawfly resistance related to, this might also actually be tied back to Jamie's presentation earlier, and I apologize because I was going to allow for Q&A after that and I totally forgot. So we can jump back to those researchers too, and if there's any questions on that. So Nicholas's question, I'll say that again, sorry. Is variation to sawfly resistance related to stem diameter? Are larger stems more resistant? I believe that question is probably directed towards you, Jamie, if you're still on.
So I'd have to look back. We recently published a paper about barley resistance, but I actually think that the sawflies preferentially choose the bigger stem. And so I think that I know that's, and I mean, of course, we don't want to necessarily choose small stems just to make sure, but that doesn't seem to be what the resistance is. We really don't know what the resistance is at this point. It's an area of active research for us.
Mm-hmm. Okay, thank you. And sorry about that, folks.
No, it's good.
I would just second that, what Jamie said, that it's probably not related to stem diameter, because we have evidence from triticale that bigger stems just mean bigger sawflies. So.
Thanks for chiming in on that. There was one question put in the Slido Q&A about whether or not there's pests that are not addressed in the 2022 annual report, and whether or not maybe we're missing something, and maybe it got a little bit to some of those pests that weren't answered in the questionnaire. But if anybody else wanted to chime in on that, other things that they're concerned about that maybe we're not currently addressing with the BPI, would love to hear that. And while you reflect on that, Juliet, you have your hand raised if you'd like to chime in.
I was going to ask Catherine whether or not bigger stems equals bigger sawflies. Has there been any work on the fact that palisades actually creates thicker stems, and as a result might change the susceptibility to the stem sawfly?
Yes, so that I can't say that we have an actual answer for that yet, but there is, I do think there's a difference between bigger stems and thicker stems, and that's something we're looking at more closely in our wheat projects right now, but we likely will have some barley projects starting up on wheat stem sawfly. Collaborations between Doe and Amanda Easterly out in the Panhandle of Nebraska. So that is something we'll be keeping an eye on.
All right, and there's a lot of good discussion actually happening in the chat on Zoom. If anybody wants to open that up and take a look. Some talk about ergot. Juliet, you also mentioned wireworms, covered smut, and soil-borne pathogens are still issues. Those were 3 that were identified as being ones that nobody responded to in the questionnaire. So certainly some support there to keep those in. Also conversation around ergot being a concern, especially with more cover crops and hybrid wheat. Also, just a comment about Hessian fly. Is the Barley Pest Initiative interested in funding those projects? And I'm certainly not the one to answer that, because you guys are all the researchers. You're the one doing this work, and you're also, you know much better what's happening on the landscape and whether or not we should be working on some of these things. I will say that the way that the Barley Pest Initiative was kind of originated, there are allocations of funding to go to each of the locations, and it's certainly really up to those locations to decide how they want to spend those funds on the pests that they feel are most important, kind of the most emerging issues. So I'll just kind of share that we're certainly open for you all to go different directions or certainly work on those emerging pests if they come about. And sounds like Hessian fly is becoming an issue in barley. Juliet's reporting. Right. Anyone else that wants to chime in on this discussion? Again, I'm not proposing we make any substantial changes in the BPI, but I just thought it would be enlightening for folks to see really how those funds are currently being spent and on what. And then hopefully that'll just kind of inform maybe your work or how we can refocus. And I do hope to run the questionnaire now each year, and we can talk too about how to improve that in the future, since that was just our first run at it. But I do think that this information is helpful. And again, it helps me when I go to Congress and I'm able to tell these stories and sharing that information on what research is happening. So,
Ashley, is there anything that you need from our group in terms of the lobbying effort as you prepare for that?
Yeah, so I'm in the process of actually putting together the packet. For those of you that have lobbied with NBIC before, very familiar with the Mike Davis packet that gets carried around that helps kind of tell that story. I'm working on that currently for our next lobbying effort, and I may be reaching out. I have all of that awesome information you guys already sent me through the questionnaire, and trust me that I'm actually working through that and trying to pull some kind of nice little stories or snippets of success that I can share. But if I run across something that I want to expand on a little further, I may be reaching out to get some clarity on that. But ultimately, I'm hoping that that questionnaire each year is what I use to help inform that packet work. So thanks for the question, Brian, but I think right now I'm set, but I may be reaching out. And also I really appreciate all the pictures that you guys submitted through the questionnaire because I'm always searching for good pictures, especially of disease, damage, or work, research, actual, hands-on stuff going on in the field. And so having that content and those pictures are really nice, again, to tell that story. So. All right, so I'm going to keep moving along. Again, I just really wanted to open up a conversation for folks to be talking about these issues. Certainly, again, don't mean to suggest that we need to make any changes in the way we're doing things, but just want to make sure everyone knows kind of what's happening, what's going on, and then if there's maybe some interest with some of these emerging pests that certainly would welcome collaboration for folks to be working on that together. So I wanted to leave a little bit of time at the end here just to talk, because I've got so many researchers on the call today, about the new initiative that the National Barley Improvement Committee is going to lobby on behalf for. And I should have said this at the beginning, but the National Barley Improvement Committee, for those folks that maybe aren't very familiar with it, is a coalition of partners across the entire barley supply chain. We get farmers, we get researchers, and we get end users of barley, and all barley types beyond just malting barley, but also food and feed. We go to DC each spring and lobby it on behalf of various initiatives that primarily are focused on barley research funding. And so the U.S. Wheat and Barley Scab Initiative and the Small Grains Genomic Initiative have been two very kind of important initiatives that have generated research dollars for barley. More recently, we've been lobbying on behalf of the Barley Pest Initiative that we're talking about today. But as we hopefully get kind of to the finish line, or the funding for the BPI, we really want to have kind of a new initiative on the horizon. And a big thank you to folks both within NBIC and kind of beyond, some other supporting researchers. We've started developing a new initiative around resiliency, and specifically thinking about how do we become a more resilient crop in the face of a lot of abiotic stressors. And so what we're hoping to do is develop a very similar initiative in the way that it's formed to the BPI, where it would have a structure with both ARS and land grant researchers. And really the purpose of this work would be focused on the need to build greater resiliency within our barley supply chain. The year 2021 really highlighted how kind of fragile, you could say, our barley supply chain is, given the extreme drought conditions that the crop was in, and then also some challenges we had with pre-harvest sprout, even in those areas that maybe weren't as drought stressed. And so really focusing on building resiliency within our barley crop so that we can have a more sustainable supply every year. So this spring, next month, we will be going to DC, again, hyper-focused on the Barley Pest Initiative. But we do want to introduce the idea of this new resiliency initiative and then plan to actually have a financial ask for the initiative in FY25, so in spring of 24. So I did want to share just a little bit about what this initiative will look like. We really plan kind of 2 tracks of work. And again, hoping to engage many of our land grants and ARS sites. And many of you guys I've already had conversations with on how you might be able to be involved in this. And I know there's a lot of conversations yet to be had. So thanks to those of you that have been a part of this thus far. But the first bucket of work that we really want to focus on is our breeding efforts. And so many of you have been engaged in this previously. And of course, we know that building resiliency is going to mean developing better varieties. And that's really been a capstone of the NBIC lobbying efforts, is to get barley research funding so that we can do more of our public breeding. And so what this work is going to focus on is investment in those barley breeding programs so that we can continue to improve genetics to really provide better resistance to those stressors. And I will note that through this resiliency initiative, there is a very strong focus on greater development of our winter barley varieties. I'm not suggesting that all of this work is only going to focus on winter, but I think that many of us in this industry recognize that winter barley provides a great opportunity. Not only because of climate change may we be able to start growing winter barley in more areas, but also if we can get that winter crop in, we're providing nice ground cover through the winter months. And then in the spring, we might be able to get out ahead of some of these insects and disease challenges that we have, but also be able to harvest before really kind of the drought or those hot season, the hot season really sets in. And so what we're wanting to do is really invest a lot more in variety development with winter barley and see where that can take us. And so across all types, though, we are focused on moisture stress. So not just drought tolerance, which is probably the one that people think of the most, but I know that we've got some researchers on the call that represent kind of the eastern US where too much water is the problem. And so across kind of the whole spectrum is how can we make barley a bit more resilient to kind of just varying levels of water availability. Also focused on temperature stress, and again, kind of representing a broad spectrum here. So both, how can we make barley more resilient to that kind of like heat stress that experiences often during grain fill, but when we think about winter hardiness, so that cold stress tolerance, and even in the spring, when we want to get that crop in sooner, how can we make sure that the genetics in that barley are going to perform well, maybe even under very cold kind of environment at the onset. We also want to focus breeding efforts around sprout challenges. So kind of thinking about how do we balance sprout and dormancy. We know that we want specifically for the malting industry, a barley that's going to come in the malt house, sprout and perform well for our maltsters, but we don't want that to happen too soon out in the field. And I know that we've been challenged with that, especially with kind of some unpredictable rains at harvest time. And so how do we better understand kind of the genetics at play there? And then finally, really honing in on that winter barley piece. And so putting a greater investment towards winter barley breeding. And specifically, the group has really talked about how can we better support the winter malting barley trial that Kevin Smith has so graciously led over the years. And many of you on the call I know are actively involved in this. But The WMBT has already grown in many locations across the country. However, the amount of data that comes off that trial is very site specific. And so how can we greater support those programs so that we can get some more consistent data coming off of the WMBT that will hopefully inform not only breeding efforts, but also adoption of new varieties, because I think that's where that really lies. And then the second half of this initiative is really about managing for better barley quality. And so this is going to kind of steer away a little bit from our kind of hyper focus that we've had over many, many years on the actual breeding work. And this is going to be looking more at cropping system trials. And so how can we better understand what those best practices are? And for this specifically, looking at winter barley utilization. So even if we have great varieties that are, you know, winter hardy, we also know that winter barley brings a lot of ecosystem service benefits, and we can actually get really good yields with winter barley, but we just don't see winter barley getting uptaken by our producers often. And really in terms of contracted acreage, at least in the malt market, we're seeing very, very small single digit percentages based on kind of the total contracted acreage, you know, versus our spring crop. And so trying to understand why winter is not getting picked up and how we can better articulate those benefits and help also develop recommendations to our farmers. And then within these cropping system trials, looking really across all barley types, is thinking about how do we optimize inputs to really kind of promote the sustainable nature of barley. So whether it be nutrient use efficiency, irrigation, so water use efficiency, or maybe even things about more cultural practices like tillage. And so within this, we hope to do some actual winter barley cropping system trials. And I'm really excited because we're actually going to try to leverage, maybe not directly, but indirectly, some of the long-term ag ecosystem trials, the LTAR trials through ARS, and see whether or not we can get barley either integrated in some of those trials or running some parallel trials, since there's already such an established framework of research, long-term research going on. And then also doing some barley management trials, trying to, again, answer those questions around how do we optimize inputs. And then finally, taking this information that we've learned and actually putting it into informed barley production recommendations. And again, this is actually pretty specific to winter because we have a lot of this content already developed for spring. A lot of you already maintain spring production guides, but that winter barley piece, there certainly are some kind of holes there, and specific to maybe where barley hasn't been grown for a while and maybe producers are starting to look at barley. I know Nebraska is one of those examples. Catherine's mentioned how, you know, they've got people that would like to produce, but those production guides really haven't been updated in years. And so how can we develop those based on kind of the understanding through some of this new research so that we can really get farmers off on the right foot? And again, this would be all barley types, not just malt, because there's certainly an increased interest in both feed and food barley as well. So that is the new resiliency initiative. And again, thanks to all of you that have been engaged in the actual development of this concept. And I just wanted to introduce it since we had so many people on the call today that represent barley research, that this is kind of what's on the horizon. And if you haven't heard from me already about this new initiative and how you can be engaged in it, you will likely hear from me over the next few months. And certainly if you have ideas on how you could engage in this research, don't wait for me to reach out to you. You know, certainly if you've got some ideas, I would love to hear them and you can share them with the group. So with that, that's the end of my slideshow. But what I wanted to do, and we're actually coming up right at time, I wanted to leave 10 minutes at the end of this call just to open it up to everyone. Again, folks can either drop things in chat, put things in the Q&A on Slido, or just come off mute, is talking a little bit back to the Barley Pest Initiative item. And if there were anything that you wanted to share related to the way the questionnaire was done, or this forum, or how the BPI is currently being managed. Or maybe even you know of a researcher that's not currently getting funding through BPI that maybe should, and that we should get kind of brought into this network. I just wanted to open up some time at the end of this call just to kind of talk about all things BPI, since we don't formally get together really in any other forum. So with that, I'll just let folks talk if they want. And Jonathan, it looks like you got your hand up if you wanted to chime in.
Hey everybody, Jonathan Jacobs here from Ohio State. I think this is a really cool initiative that you're promoting around barley production. And have you engaged with any of the professional societies like APS or ASPB, where they have public policy boards that are in DC that could also advocate for this simultaneously with AMBA, where like you could have multiple points that are trying to pressure Congress to fund something like this? So have you engaged with any of them on that?
Yeah, so maybe not specifically the groups that you mentioned, but we do work with the National Barley Growers Association, and they actually will be in DC next week. Scott will be with that group, and they do try to align many of the priorities that the NBIC has set and take those to DC as well. And so the Barley Pest Initiative specifically will be one of those that NBGA will be advocating on behalf next week. I do know the Brewers Association often too will pick up some of the items that we are advocating for specific to barley research, and will use that in some of their talking points too. A lot of times they're maybe working on more kind of industry related specific things around the craft beverage industry. But I know that there's always been a pretty strong partnership back and forth between our two organizations as well. But I can't say, and Scott, maybe you could chime in if you've got some ideas on this, whether or not there's been more engagement with other professional, like plant-based societies.
There hasn't been in the past, but you bring up a good point. I think APS would be one that does have a group that goes to Washington every year. So we'll definitely share our initiative with them, because this probably aligns with what they do. ASBC does not have a presence in DC, and they really don't organize any kind of lobbying, neither does Master Brewers, but we can certainly work with APS, which is one of their sister organizations in scientific societies. So we will reach out to them. Thanks for that idea.
I've been associated in the past with the APS Public Policy Board, and one cautionary note: I think there's going to be a resistance to promoting research in some particular commodity, because the society, to kind of favor one group of members over another. So I honestly think you're better off with the commodity grower based outreach. I think it's a good idea to make these other groups informed of what you're doing. So if they're asked about it, they can say, oh yeah, that's a good group, and we have a lot of our members contributing and whatnot. So, but I just want to throw that cautionary note out there. I don't see them really grabbing onto something that's that specific of a commodity.
Yeah, I appreciate that, Gary. Thanks for that. And like you said, it doesn't hurt to share, so we'll do that.
Right.
And just one follow up on that is, so something I started last year is drafting just a one-page legislative priorities document that's very easy to share, that we post to our website. And I do try to push that out through our NBIC network, but keep it out on our AMBA page as well. And that will be one thing too, that certainly if you have a professional society that you think might have any interest, it's a very easy one-page document that you can share, and that maybe just opens the discussion. And then, if it doesn't go any further, it really was no, extra work other than a quick email. So I'll make sure that that gets out too. And Jonathan, if you have any other ideas of other organizations, that would be a good document to push, because they'll be able to tell pretty quickly if it's any initiatives that they would like to kind of work on for us. Roger, I think you had your hand up next, or was coming up to you.
I was going to mention the ASPB for the abiotic stress initiative, but then listening to Gary, I fully agree with that. They might be, I don't know how they approach things like kind of completely plant broad based. So I don't know if they're going to want to focus on one crop except for corn or something. Anyway, that ASPB has a group that goes to Washington as well.
Okay.
That was it. But I agree with Gary, that seems a little. There might be a little resistance there, I don't know.
I may have to just throw a question out to the group. For those of you that filled out the BPI questionnaire, were you cussing me the whole time? Did the logic of the form work for you? Like, how can I make that better? Did anybody have any ideas on that? I know no one wants more reporting. I get that. So, but let's move past that and figure out how we could do it better. Nicholas, were you going to say something?
Yeah, I just want to say I thought it was great. I mean, I was happy to, I thought it was the right length that allowed a level of detail to be put in there, but was also something that, you know, I don't think I spent more than an hour or two on it. So it wasn't this huge, I didn't think it was a huge burden at all. I thought it was actually quite refreshing, new format for reporting. So
Actually, Ashley, I need to go back and look at the form because it was a while back.
Yeah.
So
It
I don't
was
remember what the
August-ish.
format was.
Yeah.
But I do remember, I thought it was a good idea to actually contribute what was happening at the location level, at least.
Mhm.
Because that has never come out before this time.
Great.
So I think it's a good idea to engage the locations to at least let them know, or let them know that they have the ability to communicate what they're doing.
Well, thanks for that feedback.
But I need to go back and look at what I said or what you asked to know if it was a.
Yeah. No, that's
Easy
totally
enough.
fair. It's been a long time ago. And
Yeah.
you'll also see in that annual report that'll go out in this at the follow-up email after this. You'll get that document and you'll kind of see how I've organized the information. I tried to kind of distill it down because I got a lot of great feedback from all of you. And so hopefully though it's all helpful. But I would love some feedback too on how I can make that annual report more useful for you. All right. Any other items?
Sean here.
Yeah.
Actually, I think you mentioned the BA, the Brewers Association is doing that. I think they do that quite actively. On the call here, actually, Chuck Skybeck is from BA and also some other members like Scott Dorsch. I don't know their effort in Washington was more on the taxation and regulations, etc. But I think it would be good for them to come back to the supply chain and overlap a little bit with us.
Yeah, and I know that at least if Chuck still is on the call, I know the supply chain is at the root of what he does every day. So certainly, and likewise, we also try to move initiatives forward for our end user industries. And primarily that's been beer in the past, but certainly welcome to try to help out with that anytime we can too. So.
I actually have a kind of a naive question.
Yeah.
For those of you that go to Washington, I have no idea how you address these problems or what you'd say to Congress. And I'm especially interested now because of the BPI, how they react to all these diseases and the problems that any plant scientists face.
Mm-hmm.
You know, do they understand that's a problem, or do they think that's not a big issue for them?
Yeah, so I can just, if anybody else on the MBIC that wants to chime in that's experienced, feel free to. But for us, you really have to kind of boil it down to economic impact. Specifically, depending on what state office you're in, the impact to their state is also helpful. And then why the BPI I think has been so effective is because it is in so many states. And so when you can actually say X amount of dollars is going to come back to your state through research funding, those are kind of the things you talk about. To actually get into the nitty gritty of any one disease or insect pest, they'll glaze over very, very quickly. So it's more about kind of very big picture, high level. This is a problem and it's costing your farmers this much money. We want X amount of dollars to fix those problems. And look, now that money's getting invested back into your state. So that's really the narrative. And I see a couple NBIC members at least nodding their head, but that's essentially the narrative that gets played out in the room. And so we don't talk about the actual insects and diseases very specifically.
One of the things I wanted to mention, mostly because that's what I do, but is the BPIs in a lot of different states, but those other states have other crops in them, and the BPI actually affects, by the research they do on barley or barley disease, their particular disease, that disease might be present in their other crops as well.
Mm-hmm.
And so if there's a way you could communicate to them that the BPI affects all of the diseases in all the crops, just by the research going on, it's, because, APS or ASPB, they all have this interaction. So they have this cross linking. None of us work in a vacuum. That's what I'm saying. So, do they understand that it's not just barley? It applies to to actually farming in their state.
Yeah, and I would say that actually, and I just glanced over to the chat and the no barley, no beers in there. Actually, that's one of the kind of the tones that we try to set is about too, that it's not just about barley and barley farmers, it's about those end uses of those products. And so specifically, I'm getting to your answer there is that the impacts in the field impact the beer industry in this state, this state, this state, so that it is more of a national problem. This is not just a barley problem in Idaho, right? Because there's a lot of barley farmers in Idaho. So we don't necessarily, at least I haven't gotten much into that like explanation of how this disease impacts, these other crops in these other states. But we do talk more about the actual kind of value added impact that if we don't procure our barley here locally and we don't have that supply, that has kind of a detrimental impact on all these end users as well, because a lot of the folks on the NBIC do represent those end user industries, and they can speak very well about when I don't have my barley, then my industry doesn't thrive. So I see your point, Roger, and that would be something probably to think about and how we can communicate that better in some of these offices where we don't necessarily have a barley footprint. And that's a lot of them actually. So that might be something to think about. And if you have any ideas on ways to kind of translate that, I mean, specifically when I go to Joni Ernst's office in Iowa for Iowa, Roger, what else could I be talking about, right? I mean, fortunately, I was getting some of that money through you. To tell you the truth,
I don't have a clue.
Yeah. But it is hard to talk to that office about barley because that's just not grown in Iowa anymore.
I know, but corn is.
Yes.
And corn has as many fungal diseases as barley does.
Mm-hmm.
And so the work done in barley actually affects research in corn or soybeans.
Yeah.
In a big way. Because, like I said, it's all cross fertilizes the field actually cross fertilizes each other. So, no pun intended. But yeah, so like I used to work, I still do, I work on barley and corn and wheat. Mm-hmm. And so everything cross fertilizes itself, in the lab, in the project and all the outcomes. So
yeah.
Joni should understand corn and soybeans, and you could talk to Michelle about that as well. I don't know. I'm not sure how much I'm allowed to say.
Yeah. Chuck, do you have something you want to say?
Yeah, I'll just echo some of the comments you said, as well as some of the comments in the chat. And, Juliet's noting that, one of the things that the Brewers Association can bring to the table is that we can usually talk about, I shouldn't say usually, always talk about a constituent business in their district, because there are breweries in every congressional district in the United States. And I know with this group, probably beer doesn't quite rate up there with pests and diseases, but certainly with members of Congress and their staff, sometimes beer trumps disease. So.
All right, well, we have already gone past our time. But I just want to say thank you to everyone that came on today. I saw at one time we had 67 people on, and that just, I'm so excited that people showed up today. And I really appreciate the time that you spent, not only as a researcher, if you filled out that questionnaire, but also just spending with us today. And I will be sending out that email that has a survey link to it. And I just want feedback on this forum and whether or not this is something you want to continue doing just once a year to kind of touch base. For the work that we're doing at the BPI. I think each year we're going to have more and more exciting things to share. We're very kind of like at the early stages of getting some of this work launched. So I think this forum could become more and more robust down the road, and maybe we might even have to find a reason to get together in person sometime. But just want to say thank you to everyone that's either collaborating or supporting one way or another the Barley Pest Initiative. And with that, I'll go ahead and close it out for the day. So thank you, everyone.
Thank you.
Thank you.
Thank you.